Hi everyone. In today's video, I'll be going through the process of meiosis. This is a particular cell division process that only occurs in special cells, and these are what we call gametes. These cells are important in the process of reproduction, unlike mitosis, which is a process that occurs in somatic cells and is more so associated with growth and repair. Meiosis, therefore, is because it's involved with such an important process, it is more complicated than mitosis. And in this video, I'll be going through both stages of meiosis, meiosis 1 and meiosis 2. So let's first start with meiosis 1, which is the bulk of the entire meiosis process. So meiosis 1 starts with prophase 1. And prophase 1 is one of the most important stages in meiosis. Not only does it take up 90% of the total time that meiosis takes place, but it is what introduces genetic variability. So, what actually happens in prophase 1? If we have a cell here, and let's say that the nuclear membrane and nucleolus have already broken down, and we only have the chromosomes present, just to make it simple. So say we have 4 chromosomes on this side, 4 on this side. So this process is unique because let's say, for example, that these 4 chromosomes are from the mother and these 4 chromosomes are from the father. And what happens is that the chromosomes that are homologous or complementary to one another are able to be brought towards one another and undergo a special process called crossover. So let's zoom into this part of prophase. So taking, say for example, these 2 chromosomes, what they will do is that first off, they will start at other sides of the cell, and eventually they will come together and form what is called a tetrad. A tetrad is essentially 2 homologous chromosomes joined to one another with what we call a cohesin protein. And this protein is important because not only does it allow for the cells to join to one another, but ensures that during anaphase, they are able to properly separate from one another and not cause for non-disjunction to occur, which I explained in a previous video. So once these homologous pairs or tetrads have been formed, they undergo crossover. And I'll draw the chromosomes a little bit bigger for this. What essentially happens during crossover is that the 2 homologous chromosomes are able to swap complementary genetic material. So if we have our tetrad here, say for example, I'll condense it down to just maybe this arm of the chromosome on, let's say, the father, and let's have this arm of the chromosome of the mother, just to make it simple. So if we have this arm and this arm crossing over, the point at which they cross over is quite important because it allows for the swapping of complementary genetic material. Say, for example, your father has a particular genetic trait that allows him to have dry earwax, and your mother has wet earwax. What will happen here is that that particular earwax gene will be swapped over or will have genetic variability introduced to it in order to have a specific trait in the fetus. So, and the point that the 2 chromosomes cross over one another has a special name. That is that it's called a chiasma, or plural, it's called a chiasmata. The chiasmata are the points at which the complementary genetic information is swapped, but it's also important once again in anaphase because it ensures that the chromosomes are able to separate from one another properly. And if you've noticed, there have already been 2 different mechanisms in order for the splitting off of the chromosomes to occur properly, because this is very, very important, because if they do not split properly, then there can be quite dire consequences for the fetus, because they can result in a whole range of genetic mutations. So once these chiasma have been produced, if we go back to this cell here, what essentially occurs, if I redraw it down here, is that new chromosomes have essentially been formed, because if two chromosomes swap genetic material, they're not the same chromosomes anymore. So what happens now is that the microtubules, similar to mitosis, are able to start to move the chromosomes towards the metaphase plate of the cell so that they can undergo anaphase. And this metaphase stage is once again important because it is able to allow for the identification of genetic mutations in both babies and in adults, because this is the point at which the chromosomes are at their most condensed stage, and geneticists can make karyotypes which are able to map out the genes of a particular person and try to identify any specific genetic mutations that may be present. But in terms of meiosis specifically, the main objective here is just to allow for the proper alignment of the chromosomes along the equator of the cell. So here we have metaphase 1. Now, after metaphase 1, the process of meiosis gets much simpler, because unlike crossing over or any swapping of genetic material that occurs, the process is quite similar to mitosis. So after this entire process has occurred, anaphase 1 can start. And in anaphase 1, the process is quite similar to anaphase in mitosis. However, instead of the actual chromosomes splitting into their sister chromatids, because we have two homologous chromosomes joined together, they instead are separated from one another. So if we have the two centriole pairs here, we have one chromosome here, one chromosome here, being split apart, one chromosome here, one chromosome here, and so on, until all of the chromosomes from the homologous pair have separated from one another. So we have this particular conformation. Now, after anaphase, we have telophase. The main difference between telophase in meiosis 1 compared to mitosis is that, unlike in telophase, when the nuclear membrane and nucleolus reform, in telophase and meiosis, the nuclear membrane and nucleolus do not reform. Instead, the genetic material that is arranged in the chromosomes essentially just relaxes a little bit. It's kind of like a stage where the cell just takes sort of a break and gets ready for the next stage of meiosis. So if we draw the cell, obviously it's going to get larger now because it's preparing to divide. And in telophase, the chromosomes somewhat relax, so I'll draw them a bit bigger than what they are in a more disorganized structure. And there is no reformation of the chromosome or the nuclear membrane in any manner. And finally, in cytokinesis 1, similar to the process of cytokinesis in mitosis, there's the formation of a cleavage furrow, and eventually the formation of two daughter cells. And the unique thing about these daughter cells is that they are all genetically, or they are both genetically different to the parent cell that once was present, because crossing over has taken place. Another quite important stage that I forgot to mention actually during metaphase is that the process of independent assortment occurs. Independent assortment is quite important for genetic variability, because essentially it means that the cell can introduce another layer of genetic variants. So as the chromosomes align along the metaphase plate in the cell, there's no predefined or particular structure that they have to have. The mother's chromosome can be on top of the father's chromosome or the other way around. So that means that essentially the chromosomes are able to have a particular genetic variability, even based on the way that they're arranged in a particular manner. So that's essentially what occurs in meiosis 1. The net result of meiosis 1 is that there is a formation of two daughter cells that are genetically different from one another and from the parent cell. Now this leads us on to meiosis 2. Meiosis 2 is quite a simple process because it's quite similar to mitosis. Unlike meiosis 1, where we went through this entire process of genetic variability, there is no need to introduce any genetic variability here because it's already been done. So essentially in meiosis, the process is identical to mitosis without any genetic duplication. So we have these two daughter cells that have been formed from meiosis 1, and if I redraw them here with their 4 chromosomes, what essentially occurs is that in prophase 1, which I'll abbreviate to P2, the kinetochore microtubules and microtubules will start to form from the centriole pairs on the both poles of the cell, and they'll essentially start to bring the chromosomes towards the center of the equator of the cell, which is, as I've mentioned quite a number of times, is the metaphase plate. So as we can see here, the chromosomes are now being pulled by these microtubules towards the center of the cell. Then in metaphase 2, which I'll abbreviate to M2, the chromosomes will all be arranged along the metaphase plate. I won't draw the microtubules and centrioles just for this stage because I've already drawn them. And finally, in telophase 2, which I'll abbreviate to T2, anaphase 2, the chromosomes will separate from one another, and the difference that occurs here is that the process is identical to mitosis, in that now, if I redraw the centrioles, the sister chromatids will separate from one another, and they will essentially form chromosomes of themselves, because once the sister chromatids have separated from one another, they're no longer called sister chromatids. They're simply called chromosomes. And finally, in telophase 2, now the nuclear membrane and nucleolus will reform. So here we'll have the 4 chromosomes present with the nucleolus, and similar to telophase in meiosis 1, the genetic material will start to relax. And finally, in cytokinesis 2, the chromosomes, the cleavage furrow will form between the two cells, like so. And we will have the formation of 4, in addition to the 2 that were there before, genetically different haploid cells. So I'll just redraw the nucleolus, because as we know, there are 2 nuclei present now in each cell, and that will mean that eventually there's the formation of 4 genetically different haploid cells. And these cells are genetically different to one another, whilst also being genetically different to the parent cell. And that is the end of meiosis. Essentially, these are the cells, whether they be spermatozoa or oocytes, that will come together and fertilize in order to form the zygote and eventually the fetus, which will give us a baby.